Imagine a world where a simple shift in perspective could unlock new possibilities for treating a deadly disease. That's exactly what researchers at Penn State have discovered, and it's a game-changer for leukemia patients. Two drugs, apalutamide and finasteride, commonly used to treat prostate cancer, have shown promising results in fighting acute myeloid leukemia, AML. This is a significant breakthrough, as AML is a challenging cancer that affects the blood and bone marrow, and it's the most common type of leukemia in adults. The research, published in Blood Advances, tested these drugs on experimental models, including mice with AML and patient-derived AML cells. The results were remarkable. Both drugs effectively suppressed the progression of AML in both male and female mice, indicating their potential for broader use. Dr. K. Sundeep Prabhu, a lead author on the study and an immunology professor, explained that these drugs work by targeting a specific pathway activated by dihydrotestosterone, a potent androgen hormone. While androgen signaling has been extensively studied in prostate cancer, its role in AML has been largely unexplored until now. Our study highlights the potential of these drugs for new applications, Dr. Prabhu said. It's an exciting development, especially considering the limited treatment options for AML patients. Professor Robert Paulson, a co-author on the paper, echoed these sentiments, emphasizing the need for new treatment directions for AML. AML is a complex disease, and many patients experience relapses. Identifying a new target like the androgen receptor, for which we already have FDA-approved drugs, is a significant step forward. We're eager to see the impact of these drugs in clinical trials. But here's where it gets controversial: the connection between leukemia and androgen receptors was previously unknown. Fenghua Qian, a senior scientist at Regeneron and a lead researcher on the study, discovered this link while working on an AML mouse model. Qian's initial hypothesis was that estrogen might protect female mice from AML, but further investigation revealed that mice without ovaries, and thus no estrogen, still had a lower chance of developing the disease. This led him to explore the potential role of androgen, and his findings were groundbreaking. I analyzed female and male donor leukemic cells and found that androgen receptors were highly expressed in female donor leukemic cells. This interaction between androgen and its receptors could be a key target for leukemia treatment, Qian explained. In their study, the researchers found that high levels of dihydrotestosterone in male mice promoted AML development, despite low androgen receptors in their leukemia cells. Conversely, female mice, with low dihydrotestosterone levels, had high levels of androgen receptors in their leukemia cells, which offset the low dihydrotestosterone. Finasteride slowed AML progression by inhibiting dihydrotestosterone, while apalutamide targeted a slightly different pathway in androgen receptor activity. Qian emphasized the importance of this discovery, as it provides a novel targetable pathway for leukemia patients. Androgen receptors and their interactors could be direct targets for treatment, offering potential benefits to patients resistant to traditional therapies, he said. While the study is promising, the researchers emphasized the need for further testing, including clinical trials on leukemia patients with specific criteria, such as increased androgen receptor expression in leukemic cells. Dr. Prabhu highlighted the importance of attention to detail in scientific research. Qian's vigilance and keen observation skills led to this breakthrough. It's a perfect example of how small details can lead to significant scientific discoveries. This research opens up new avenues for leukemia treatment and offers hope to patients facing this challenging disease. It's a reminder that sometimes, the answers we seek are right in front of us, waiting to be discovered.